This is lecture 2 of the image processing. We started last time, we just talked about basic principles about digital images, how they're made, and the formats of digital images. Today we're going to start talking about intensity transformation and image histogram. So we're going to get deeper into how we're going to be processing medical images to extract information. That's the most important thing. Here is the outline, the first page of the outline. So we will be talking about what biomedical imaging is and what image processing and useful for. And then we're going to focus on image acquisition and representation. We have two ways I want to show you in which we are collecting images. And then I will be completing spatial filtering, image enhancement. We're going to enhance the images using spatial filtering, intensity transformation, and hopefully image histogram and equalization. There is an example at the end of this lecture. Next time we meet, we're going to do the convolution filters and the order static filters and do the segmentation, because this is another important thing for us as biomedical engineers. How can we do texture segmentation? So let's say you want to identify a tumor cell from normal cell, from an image from an inverted microscope, and edge detection to count cells and do active contours of cells. And then we'll do at the end what is image registration, and then we'll talk about the shape analysis. So if we completed all of these together, you should have a good idea about what image processing is. And in the lab, we're going to be doing more experiments to, use all of this into real images. Now, what is image processing for? And I'm going to show you three things, three levels of image processing. I think two of them will be including artificial intelligence. And this is going to give you an idea that we have been using artificial intelligence for a very long time in medical imaging. Now, what you're using like ChatGPT and Gemini and Copilot, those are all text-based. But we were using AI in finding all of the things that I told you before, texture, contouring, and counting, way much before. What is image acquisition and reconstruction? This is physics, math, and engineering all together in order to get an X-ray. Remember when we were talking about the systems, what is the components of X-ray and CT scan? This is acquisition and reconstruction. And so studies how to reconstruct efficiently meaningful image from the raw data. Remember when we have CT scan and we did the reconstruction of the images? That's this. And we covered that, and I hope that you are now experts in systems. Now, visualization, that's the computer graphics. That's what we would like to work with. And last time we talked about the different types of images. Now, in visualization, you study how to visualize the reconstructed image in a way that is useful for the human observer. Remember when we have the CT scans and the images, they were actually all round, like twisted images, and we said we need to reconstruct them to see the CT scan itself? That's visualization. So you start with the systems, then you do some visualization to make sure that you are seeing what you're looking at, and then we do image analysis and understanding. This is where the magic actually happens. So that's something called computer vision. So instead of doing, as I told you, I was counting cells, proliferating cells from slides. We need to make sure that the computer is doing this for us. And that's something called unobserved computer vision. So the study is how to emulate with a computer perceptual and visual behaviors similar to the biological ones. So let's say my eye is looking at the information. We would like to be able to design something that the computer can do as close as possible to biological ones. Now, studies, this also image analysis and studies models, algorithms, and techniques. So you can work, let's say, in a grad school on a project where you are developing an algorithm to do specific computer vision application for a specific set of data. You can also, you want to recognize objects, structures, and events. And I'm going to show you a number of images to get you into speed. But are we good with these? Image acquisition and reconstruction, that's the system. Then visualization, how do you see this? Reconstruct the image from different places, images. Like, the CT scan, you're taking X-ray from different angles, reconstructing them all together to get a two-dimensional scene, that's visualization. And then image analysis and understanding, that's what I would like to focus on here, and what you are going to be doing in the lab. So look at that. See here, this is our process in computer vision. We would like to focus on this because that's mainly what people are trying to do in labs, to automate. And probably I said this to you, the measure of how we are advanced in our civilization is how much automation we are doing. So you are going to acquire the signal, and you do digitization. Remember, if you have an image, you would want to be able to, so if you have a two-dimensional image, you would want to be able to do sampling from the image, and after sampling you do quantization. So you take, sampling is taking samples in the image, and then you do quantization, which means that you are going to create the levels for the illumination, if it's colored or grayscale, and then you do digitization. So it's sampling, quantization, digitization. After you have your signal as a digital signal, which is zeros and ones, you do image processing, and that's what we would want to learn. And then after you do image processing, you would like to do image analysis, and that's the pattern recognition by seeing how many cells do we have, identifying different types of cells. And then after you do the analysis, you need to do image understanding, and that's where AI comes in. And that's how we're going to use it. So digitization, we call it conditioning of the image. Then you do image processing, then you do image analysis, and then you do image understanding, and all of these together constitute computer vision. Now here's the thing, we have 3 levels of computer vision or image processing. We have the low level vision, that's what we call it, low level vision, which is image processing only. We don't have understanding, and we don't have analysis or understanding. So image processing processes set of operation performed on images, aiming to enhance their quality and selecting useful information, which will be processed by human or other algorithms. So that's the low level image. So vision, you have an image, you do a processing, and you look at another image. But you still here at this point, you need a human or an algorithm to analyze things. So it's not fully automated. It's doing processing, but you still need to look at it to make a decision. Let me show you an example. We can start with this image, right? And you have the washers and screws. You can do, if the image is blurry, it's get Gaussian, we can blur the image to get Gaussian blur, and then you do contour extraction. The Gaussian blur is an important step to get the contour extraction, and after you get the contour extraction, you do something called contour selection, in which you are removing these weird areas. And after you do contour selection, we do something called segmentation, and you fill the areas with a color, and then you can distinct between them. At this moment here, this is where it ends. That's where the low level vision ends. And then you need a human to look at the image to make decisions, also to make understanding and to make analysis. So that's a cascade of the low level processing or low level vision. Then we went a step further. We would like to do analysis, right? So we did something called mid-level vision. That's image analysis. So it includes extraction of symbolic information from pre-processed images and analysis techniques of the visual characteristics of the object. So you have an image, you do analysis, then either you are showing the image for someone else to do analysis, that's what we did with the low level, or you do features vector of the segmented object of the scene. So you want to automate the analysis one more time and to include that step. So let me show you an example about this. So let's say you have the same example of the low level, but after getting this image, you want to create an algorithm that decide which one, or characterize the objects by which one is not washers, and bullets, right? So what you do is that you are creating an algorithm that's going to measure the area of each object and measure, there is a parameter called circularity, how circular that object is. So see this one here, the long one, that's going to have a very low circularity. The nuts will have a larger circularity. Make sense? So these circularity and the area, those are function in the MATLAB toolbox that you can use, and you can create a small code, and it's going to create for you an x-axis where the areas are and the y-axis where the circularity. And you can see now you're not looking at the image itself to analyze it like the low level, but now you have a table. So the computer created a table for you with extracted features. And now then you can make much better decisions. So here, look at this. See those circular? You can see that they have high area and low, mid circularity, and you know that these are the 2 and 5. And the ones here, you can see the triangles are the nuts because they have low area, but they have higher circularity, and you know that these are the nuts. The one low circularities, the squares, are the washers. So your computer, your algorithm, is creating this image for you to analyze. So we still need you to look at the extracted information. Does that make sense? So the difference between the low level and the mid level computer vision. Now, the high level computer vision is that now you need the computer to make a decision which one is which. And what we do here is that for the high level, we need to do image understanding. So aims at obtaining some comprehension of the observed scene as shape recognition, or a spatial relationship among objects. It includes high-level abstraction processes. Now, these are the 3 things that if you really want to do medical imaging, you do classification, identification, and localization. If your code is doing this, which is making a decision here, then you are dealing with a high level of computer vision. And if you look at our example here, the computer will make a decision by creating circles, by seeing the localization, the area, or the classification, that these 4 are nuts, these 4 are washers, and these 2 are bullets. And then it can give it different colors, and you can identify them. So that's called clustering, which means classification. Non-supervised, it's by itself, makes that decision. And that's AI actually in medical imaging, and that's how advanced actually, and that's been happening for a long time. But these algorithms are only for medical imaging and medical applications. So let's say you are joining a research lab for your graduate studies, that they are interested in lung tumors, and you are going to develop an algorithm for them to analyze and to identify the size, location, distribution, and malignancy of the tumor, just from images. Does that make sense? So this is what are we using medical image processing for. So it's either low level, mid-level vision, or high-level vision. Are we good with this so far? Any questions? Okay. I will move on to the next section of the lecture, which is the digital image acquisition and representation. I want to explain 2 things quickly for you. I think, 2 things. So how are we acquiring and representing images? Just to make sure that we are on the same page. I know that we have covered this before, but just to be on the same page. So here, we have something called sensor array. This is your model, way of acquiring data. In the sensor array, you have illumination, which is light, reflected from a scene into an imaging system, like a CCD sensor, the one that we talked about last time. And you have an image system with lenses, right, to focus the light into the CCD sensor, and then you have a digital image. So this is the scene element. This is your imaging system. You have an image plane, and then the output is going to be sampled and digitized. Remember, we have a spatial geometry, independent variables, X and Y, and then we have the illumination, for each pixel as the dependent variable. And that's how you create an image, an output digitized image using a sensor array. Good? That's one. The second one is similar to what we did with the CT scan, that you have sensor strip instead of sensor array, and that's the strip here, the the circular, the sensor ring, and you have the object that's sliding in just like the CT scan. And let's say this is your imaging area, and we have the sensor strip. So either the sensor strip is moving or the object is moving below the sensor strip, and you can create through image reconstruction, just like the CT scan, cross-sectional images, two-dimensional cross-sectional images over three-dimensional object, right? And that's sensor strip. So you have either this one or the one that we described before in order to get you how to acquire an image acquisition. All right, are we good with this? Fine. Let me now go talk about sampling. And I know that we touched base on it in the last lecture. Remember that sampling produces continuous values in x and y, because x and y are the your independent, right? Spatial, multivariant, independent variable, and in each pixel, when you identify the location, there is the illumination, which is the grayscale level. Now, let's say this is your original image. When we say we would like to make it digital, we need to sample it. So you will be, if you see this here image, this one in this box, we can't have half of it white and half of it gray. What we are doing actually is we are assuming that the entire pixel have the same value. And that's what we mean by digitizing the pixel amplitude. That's quantization, because you're going to select different levels for each pixel. So if this is real, it has to be translated into this box here, where the entire pixel has the same grayscale level. So that's called pixel amplitude, and in digitizing pixel amplitude quantization. Now, n bits per pixel, how many bits are representing a pixel? This is 2 to the power n. So if you have 8 bits to represent a pixel, that's 2 to the power 8, and that's 256. And if you go from 0, including 0 as one level of quantization, you go from 0 to 255. You end up with 256 levels, right? 55. So if this is 1024 multiplied by 104, you have this image. But if you make it smaller, the bigger pixels with smaller number of pixels, your image will be blurry like this, because you are filling the void in each pixel with the same value of it. Now, see this x and y? We just want to make sure that we are all on the same page by saying that the digital image representation, the result of sampling and quantization. Sampling is selecting, and quantization is the value of the grayscale. Then you end up with a matrix, right? With values of illumination in each of the x and y. We good with that? With how they present? It's just something we talked about before, but I want to make sure that we are all on the same page. Are we good into going to the heavy stuff? So let's take 5 minutes break, and then we'll come back, because I would like now to get into the image enhancement. Hopefully we cover 3: the spatial filtering, intensity transformation, and image histogram and equalization. Let's take 5 minutes break, and then I will come back to continue with that. We good? All right, stop here. Alrighty, so again, I hope that we can cover these 3 today: spatial filtering, intensity transformation, and image histogram and equalization. Remember, what we would like to try to do is to enhance an image, and I'm going to show you examples. So there's going to be a lot of pictures here, but I think it should be good. So let's start with the spatial filtering. So let's say the simplest way to enhance an image. So let's say you have this x and y spatial scene, and you have a pixel here, x and y, and that's your from your image, based on the on direct manipulation of the pixel. So if you perform a mathematical operation on the pixel or one of the pixels, that is called the spatial filtering or direct manipulation. Remember when you said we have the operator that is transforming a signal t? That's what's happening. You have f of x of y, that's your x of y. You have the value, the intensity or the illumination and or the gray scale value, and you do transformation for it, and you come up with a new value of that pixel. And that's the simplest way of doing this. So t operates on f at a specific location, and defines over a neighborhood over the pixel x and y, for example, a small sub image centered at x, y, and that's what's going to happen. So whatever usually you can't do operation on only one pixel, but it's that pixel and its neighboring, but you center it on that pixel. So let me show you an example of what does spatial filtering mean. So let's say that's you're changing the value of one pixel. Is that take the value and square it, right? So that's called squarer manipulation for it. Now, if you want to do intensity transformation, you do spatial filtering with the neighborhood size 1 by 1. In this case, g depends only on the intensity value of f of x. So it does not depend on the location, it depends on the intensity. So you're putting a threshold on that intensity, which is the illumination or the grayscale. It is completely described as a function between intensity values. Let me show you this. See these two functions. I'm going to show you how can we use a function like this to increase the darkness of pixels, or reduce the darkness and increase the brightness. Does that make sense? So I'm going to come up with a filter or a function that performs on an image. Here's the example. Bear with me. This is an image of cells. On the right here, you know the histogram example? We said, well, the x-axis, the grayscale, values, and let's say you have a very large number of dark values. So this is a linear line. Above the line, anything above the line will be dark, and anything below the line will be bright. So you're making a threshold that you're cutting it. What if I increased I added more values from the histogram? So instead of having this, I'm making more value below the line. So I'm making it more brighter. Does that make sense? It's just like a threshold. And that is going to be the cutoff. So this here, above the line, dark, below the line, white. If it's linear, I have a lot of, values that they're going to be dark. This is reducing those values and cutting them, making it more a brighter image. So that's your scale. If I want to do the other way around, if I want to increase the number of the intensity of the darker pixels, I do something like this, right? Now, if you want to make sure that you are doing a fitting, so that you are including as many of them as possible, you can do a curve like this, and then this will make a big distinction between the dark and the white, like a threshold between the two. The key here is this line, and you can, in algorithms, create these lines. That's called intensity transformations based on the brightness of the pixels. See the ones here down there? Each point on the x-axis is a gray level, correct? If you are cutting or doing a filtration by reducing the number of pixels that they have this color by this line, so instead of having the long one, now you have the shorter one, you can manipulate the image by manipulating the intensity. So you are reducing or increasing the number of pixels with that specific intensity, gray level, with this curve. So this curve is your transformation, and this is your x of t. So that's the transformation of x of t. That's the black curve here, this one. So I'm limiting the number of pixels with that specific intensity, illumination or grayscale, and that will affect how the image will look like. So I'm not, manipulating the locations, on the XY, but I'm manipulating the number of pixels of that intensity. So that's what we call intensity transformation, rather than spatial transformation, right? So I'm not going to each pixel and multiply it by x to the power 2, but what I'm doing, I'm not squaring it, but I'm limiting the number of pixels that have that value in the image through this filter. So spatial transformation and intensity transformation, that's what we did. So intensity transformation, it's contrast enhancement. Left plot contrast, stretching, darken gray value below M, that's M, be darker, and brighten gray value above M. The right plot, it's a threshold, it's a cut threshold. So it's exactly at that point, it's either on or off. So it's either black or white, and it's select the threshold of the intensity. So here, this is an example. Let's say I'm giving you, instead of the curves, I'm giving you this, intensity transformation, and that's your image. If you apply the transformation to this image, the darker will be dark, and then the whiter will be white, and you get this one. If you have the threshold as this here, like this, it's black and white. This one here is giving you that. So this black line is given this. If I have this threshold as on and off to give you the black one. And this is based on intensity. So the x-axis here is the grayscale level. So you will select at which grayscale level you want to have the cutoff with them. So intensity A, that's the intensity transform, and that's, to me, is T. This is X, and here T of X. That's the operator or the transformation. If I have that one that is like this, cutoff, you get black and white. Now, in MATLAB, you will have functions that will do spatial transformation or the intensity transformation. Make sense? Now, we have also another, so this is contrast, this is good for contrast, the intensity transformation is good for contrast enhancement. So I'm enhancing the image. So this is not that good. This is good. We can tell what that is. Now, there's another application for it, which is doing the negative. This is a mammogram image. Usually it's going to be, the background is going to be black. If you do the negative, you can see much clearer where the tumor is and how it's spreading in the mammogram. So that is another, application of your intensity transformation for it. So equivalent of photographic negative, enhance white gray or white gray detail embedded in a black ground. Good? Let's have a look at that. So you will be looking, now if you work in a hospital, your technician, imaging technician, will be taking these images, but you need to be able to calibrate to make sure that they are doing what they're supposed to be doing with the imaging, with the images that you want to do. Are we good with that? Okay, let's go back to the histogram. Now, see this histogram. Histogram is an XY representation. The x-axis is the grayscale level, and the y-axis is how many pixels do you have in that grayscale. Is that good? Does that make sense how we do that? Now, if you look at this one here, you can see it's dark. So as closer to the left, the smaller the values, the darker the image, because you have very dark pixels. The brighter the image, they go to the right. So you have higher numbers. And remember, you go from 0 to 255, right? and in each one of them you have how many pixels in the image has that grayscale level. It's not spatial, it's not just counting, accumulation, cumulative, that's what we call it, images. So the histogram, this discrete function that associates the gray level are, the gray level are to the number of pixels having gray values of equal to r. Are we good with what's histogram of an image? Now, this is if you have a clustering of that image of the histogram, that means that your image is blurry and need enhancement. That's why we do something called histogram equalization. You want to have your histogram distributed all as wide as possible across the spectrum. This way you have a better image to look at. And that's what's happening here. So if you have this blurry image, they're all clustered in this location. Histogram equalization, this is give you a high contrast image. Low contrast, that's bad. Remember, we are looking at images in different way. In the lab, we looked at the free transform, and it was in the middle, which is the low frequencies, and the one outside are the noise. Now we're looking at a different representation of the image using histogram. If the image is like this, without looking at the image, you know that this is blurry. If it's distributed like that, you have a high contrast image, right? Now, we will learn how we can transform an image like this here, a low contrast image, to a high contrast image using something called histogram equalization. And hopefully we'll have time to do the exercise together. Make sense what we're doing? Again, another way of looking at the image to extract information and to do processing. So, histogram equalization, an enhancement method based on intensity transformation. It's intensity transformation and not spatial transformation. You find a function as strictly increasing so that when you do the transformation on F, you get a new image G, but with a spread histogram. Has uniform normalized histogram, and then you get your values. Here's what you want to do. If you're getting this one, you want to, if you do equalization, you get this one. Also this one, you'll end up with that one. So low contrast, high contrast, low contrast, high contrast. That's what we would like to do. So that's an example. This is another example that you need to move from this way to this one. You go 1, 2, 3, and that's the end results. Now, let's assume, here's what I want you to assume. Let's assume that this is a matrix, right? And that matrix has pixels. Each one of them is 1 pixel. And I'm going to represent each one of those with a number. So let's say this is 4, this is 4, this is 6, 7, 2, 1, whatever the values are. So that's your matrix. We would like to learn how can we do histogram equalization on the numbers, the pixels, by hand before using MATLAB. Does that make sense? How we're going to do that? So I will give you a piece of paper, and we would like to do this example. So let's say this is an image, right? And that image has these pixels. We good? And I want to show you how we can do the equalization. Let me give you a piece of paper. Oh, hopefully we have that. I think we have that. And let me share. Okay. I want you to do this with me because that's important. How are we going to do the equalization?
Are we copying that on our piece of paper?
Yes. Yeah, I think we have 15 minutes. I think we can do this. So I need to do it myself. So 4, 4, 4, 4, 4, 1, 2, 3, 4, 5, 3, 3, 3, 4, 4, 5, 5, 4, and then we have 5, 5, 5, 3, 4.
Are we drawing them like with the lines and boxes and all that?
No,
Or
no,
Okay.
you don't have to do this. You have the numbers? Okay, so let me show you what I'm going to do step by step. Step 1, what I will do find the probability distribution, which means I want you to draw. Before we draw this, what is the largest number we have in the pixels? It's 5, right? What is the closest 2 to the power n or n that we can use that can give us 5? So 2 to the power 3, how much is that? 2 by 2, 4 by 2, that's 8, correct? So I need to use 3 bits to describe that image. So keep that in mind, 3 bits to do the representation. So to do the probable distribution, I'm going to be creating 0. So I need 8 digits, correct? So 0, 1, 2, 3, 4, 5, 6, 7. Those are 8, correct? You draw a line here, and I would like from you to show me how the histogram will look like. So how many zeros do we have in the image? In the pixels, how many zeros? 0. You put the 0 here. How many ones? We have 0. How many twos? We have 0 twos, right? You can see that they are not distributed, correct? Already where you can see that. Now, how many threes do we have? So we do 1, 2, 3, 4, 5, 6. Right? How many fours do we have? $12. So that should be twice, correct? How many fives? 7. So that is 6. We add 1 more. That's 7. How many 6? 0 and 7 zeros, correct? So you can immediately say or tell that we have a low contrast image because it's not distributed on the entire spectrum. Make sense? So that's the how we're going to do this simplest form.
So that's low distribution.
That's a probable distribution. Now, what you need to do is this. Are we good with this one? Just to show you that it's a low contrast image. Now, you delete all of that, and I would like to create a table. Now, see this distribution that we had, me and you? Keep it there, because we would like to create one that is much more distribution. I'm going to show you how to do this. So the first thing you're going to do is you do the 0, 1, 2, 3, 4, 5, 6, 7. Correct? Now you need to tell me how many of these do we have. So the number of the grayscale here, we have 0, 0, 0, 2, 6, right on 3. We have 12 fours, 7 fives, 0, 0. Are we good? Make sense? Now, here's what you need to do. You need to find the summation of all these grays. How many do you have in the grayscales? So how much is that? 25. You're going to create something called probability distribution function, P, D, F, by dividing each value here by the summation. So 0 by 25, right? 0 by 25, 0 by 25, 6 divided by 25, 12 divided by 25, 7 divided by 25, and that's 0, 0 oh, sorry. 0, 0. Are we good? This is called probability distribution function, which is the number of pixels those are the number of pixels like that they have the grayscale. So that's a grayscale number of pixels, and that's your probability distribution function. Now, after you do this, what are those values? Can you help me with these? So let's say 0, 0, 0. How much is this one? 6 divided by 25.
0.24.
And 12 by 25?
0.48.
And 7 by 25?
0.28.
0.28. Thank you. I have 0, 0. We good? Now, after you do the probability distribution function, we find something cumulative distribution function, CDF. And here's what you do. You start with this one, you put 0. Then you add this to that, and you put the answer here. That's 0, correct? We are accumulating, we are adding cumulatively. Then you take the 0 added this one. What do we get? 0. 0 plus 20.25, 0.25. Do we agree? Then you add 0.25 to 0.48. How much is that? 0.73. Now you add 2.28.
1.01.
0.1.
Add to 0.5.
That's the
0.25. Oh, I'm sorry. Good catch. Good. Thank you. Thank you. That's 0.25. Are we good with this one? Sorry. Thank you. That makes me feel happy that everybody is paying attention. Good. Now, 0.73 plus 0.28. Is that good?
Yeah.
Now, this plus this is 1.01. This plus this is 1.01. And you ended up with cumulative distribution function. We good with this? Now, we need to do another step by multiplying by the maximum gray level 3 bits, which is 7, right? We said with 2 to the power 3 is 8, but I need to go from 0 to 7 to have 8 levels, 8 quantization levels. So you multiply each one of those by 7. So that's 0 multiplied by 7 is 0. That's 0. That's 0. How much is 0.25 multiplied by 7?
1.75.
7.5. How about 0.73?
5.11.
And 7.01?
Oh,
Oh, no.
5.07.
Or 7. Is that correct? And then 7.07 and 7.07. We good? That's the multiplication. Then you do approximate, you do rounding. So 0, 0, 0, that's 2, that's 6, and then 7, 7, 7. Make sense? We good? Right? X-axis with the approximation. Yeah. So here's what you're going to do. See, we have 0. So we need to draw our histogram, correct? So at 0, we have 0. At 1, we have also 0. At 2, we have also 0. At 3, instead of 6, what do we have? 2. So that's 1, 2. At 4, we have, instead of 12, what are we getting? 6, right? We are replacing these. So 1, 2, 3, 4, 5, 6. What do we have at 7? Instead of at 5, instead of 7, we are getting 7, right? So that is 7, 7, and 7. Now we have a much, instead of distributing it on 3 values, we are distributing it on how many values? 5 values. So you did equalization. And instead of having it on 3 only, now you have them on 5. So you have 0, 0, 0, 2, 6, 7, 7, 7. Your image this way will be high resolution rather than low resolution with only 3. So you did some equalization with that. Make sense? How we do that? Now, here's the important thing. I want you just to know the probability distribution function, the cumulative distribution function. You multiply with the largest bit that you have, the equalization level that you have, which is 7, and then you do rounding, and you replace each one of them with the values that you should have. So we moved from the blue that was, 1, 2, 3, right, into that. So that's equalization, called histogram equalization. And we have in MATLAB functions that does that for us. Because this is just, it's a very small number of pixels, but if you have 1024 multiplied by 1024, that's a huge number of pixels. We can't do that by hand. So MATLAB has the algorithm to optimize it to do histogram equalization. Now, just to make sure that we're on the same page, why are we doing this? Image enhancement. So you get high resolution image from a low resolution image with that thing. Sounds good? Okay, we have 3 minutes. Go back to the multiple choices. Double check that you have all of them, and I will give you the answers quickly. Let me do the answers for you, and just you double check with that. So what is the primary goal of image acquisition and reconstruction? It's C, to construct meaningful image from raw data. You have CT scans from different angles, you're making an image from it. Now, 2, which of the following is not a primary focus of image processing? C, emulating human perception, because that's meaningful. It's not processing. Remember, you have an image, you do image processing, you do analysis, and then you do making meaning. So that's emulating human perception is the high-level imaging, video imaging. What is a key difference between low-level vision and mid-level vision? It's B. Low-level vision enhances the image quality. Mid-level extract information, symbolic information, right? Remember when you have that chart with each one have the area and one has the circularity. Which of the following is an example of a mid-level processing feature vector extraction? Because you're looking at the area and you're looking at other things. So what does high-level vision aim to achieve? Comprehension, because you need to make a decision, right, of the observed scene. Which of the following processes is included in high-level classification? Remember, classification, because you need to make categories, right? And that's high-level. What is the purpose of the image enhancement technique? To improve the visual appearance of an image. That's all what we're trying to do. Right? Which of the following is an image enhancement technique? Intensity transformation. What does image segmentation involve? Dividing an image into meaningful parts. That's segmentation. Right? Clustering. That's what we do. Clustering. I know that I want you to know the terminology. That's important. Which of the following is a segmentation technique? Thresholding. You separating things from each other, just like black and white. That's segmentation. So what is the goal of image registration? To align or match 2 or more images. What is a histogram in an image processing? Graphical representation of the total distribution of an image, right? We're adding the pixels in a gray level. Which type of image processing is used to enhance the quality of an image and select useful information? Low level. It's the basic level of processing. What does image analysis involve? Extracting symbolic information from pre-processed images. That's mid-level, right? 15. Which of the following is an example of a visual primitive that can be extracted during a mid-level processing? Area, That's what we showed the example, area and circularity. What is the purpose of histogram equalization? Adjust the intensity distribution of an image, correct? You're spreading this thing around. In the processes of computer vision, which of the following comes first? Acquisition. Which of the following is not a type of image enhancement? Edge detection. That's not enhancement. We are making decisions. Segmentation, right? Which of the following is type of spatial filtering? Now we're going to talk about those in the future. Convolution filters, we didn't touch base on them, but that's spatial filtering. It's not intensity. What is the main goal of image understanding? Comprehend an observed image. Make sense? Was this successful? Useful? We learned something? Cool. Can you take it? I have week 10. Take a Dropbox. Take an image of your histogram equalization and of the multiple choices. All of them, yes. This is going to be a quiz for you. So. Any questions? We good? This side?
Great.
Awesome. I will stop recording. I'm sorry, I took